So we're going to kick things off. Hello, everybody. Welcome to this penultimate Validate online seminar of 2020. We have one more coming after this, which I'll talk about later. My name is Blakely Nixon. I'm the Events and Communications Coordinator at Validate. Many of you probably know that already because you're probably annoyed by all the emails you get from me. And before I start, I want to do some very boring housekeeping stuff before I hand over to our excellent and amazing speakers. So the main one, and I think everybody knows this by now after 10 months of lockdown, during the talks, can you please turn off your camera and mute, but only during the talks. What we're going to do today is we're going to have the talks, we're going to run through the talks all the way through. So we're doing 2 talks in a row because they're linked, and then we'll do a Q&A. So feel free to turn on your camera during the Q&A, but keep it muted unless you're asking a question. The Q&A tends to sort of manage itself and work itself out depending on the groups, but broadly speaking, if you have a question, there's a hand raise function along the top. It's just a little hand, and you can click on that, or if you want to ask a question using the microphone, or if you would prefer to sort of be a bit quieter, you can type in a question in the chat function, and I will try and spot them out for us speakers. The other really important thing is this is being recorded. And so if you do ask a question or appear on the video, you will be in our on our recording, which we'll probably upload to YouTube. This isn't a confidential talk, so we're going to be sharing this quite widely. So if you have any problems being recorded, just send me an email and I'll make sure to sort of edit out or edit around you. It's not a problem at all. And finally, if you could all stay around afterwards for the rather socially awkward bit where I ask people to wave at the camera. Because it's just a nice picture showing us all having an amazing time listening to these seminars. So without further ado, I'm going to introduce our speakers very briefly. Louise Gourlay is an assistant professor in biochemistry at the Department of Biosciences at the University of Milan in Italy, and Giorgio Colombo is a professor of organic chemistry at the Department of Chemistry at the University of Pavia. Louise is going to start, so I'm going to hand over to her, and I will be back during the Q&A to sort of manage things there. So over to you, Louise.
Okay, well, thank you very much for the invite. As you can see from my title, I'm going to talk a bit about how structural biology can be used to try and design epitope-based therapeutics and diagnostics. So as Blakely mentioned, I work at the Department of Biosciences, specifically in the structural biology unit, where we combine different structural biology techniques, so in particular X-ray crystallography, but also single particle cryo-electron microscopy, to solve the 3-dimensional structures of our protein targets, which are of biological relevance because they're linked to different human pathologies. So as a large group, we look at a number of different protein targets that range from transcription factors to viral proteins, and also to protein antigens, which is the focus of my seminar today. So the use of 3D structure information to guide the engineering of an improved antigen. So when I say improved, I mean an antigen that has better immunological or biochemical properties, has been coined as structural vaccinology. The pioneering group in this field is the group down in Siena, which was Novartis, ex-Novartis, now GSK. And I've reported 3 of their examples of structural vaccinology that are available in the literature. So if you look at the top left panel, we have an example of an engineered antigen from the meningococcal type B pathogen that was specifically engineered using 3D structure information to host epitopes from the same antigen from 3 bacterial variants. And so when this engineered antigen was used in protection studies, it was able to induce cross-protection against all 3 variants. If we look in the top right corner, we can also see an example of a viral antigen that was observed to that has 2 conformations, a pre-fusion and post-fusion conformation. And the X-ray crystallographic structures of these 2 conformations allowed the authors to pinpoint the introduction of a disulfide bond in the pre-fusion conformation, therefore locking it in this conformation, because
Yeah.
Okay, so here I was showing you the example whereby a disulfide bond was introduced into the pre-fusion conformation of this antigen to block it, since this is the form of the protein that contains most neutralizing antibodies, epitopes, sorry. In the bottom panel, we have an example from the group B Streptococcus pathogen, where actually I was personally involved in this work, and we solved the crystal structure of this antigen that revealed that it's organized into 3 different domains. Only one of these domains, highlighted in red, was known to host the neutralizing epitopes, which allowed the production of a chimeric antigen comprising 6 of these domains from 6 different variants. And once again, this chimeric antigen was able to induce cross-protection in vivo trials. So hadn't the crystal structure of this antigen been made available, we wouldn't have known that it was a multi-domain protein, and therefore it wouldn't have been possible to generate this chimeric antigen, because of course it's quite a large antigen, and this wouldn't have resulted in soluble production of this antigen in recombinant systems. So then, after this work, we moved on to try and to look at protein antigens from Burkholderia pseudomallei, which I'm sure all Validate members are familiar with. So it's a gram-negative soil-borne bacterium that causes melioidosis, which affects populations mainly in the tropical and subtropical regions of the world. As you can see in the bottom figure, the global distribution is shown. There are several routes of infection, including inhalation, ingestion of contaminated water, also through cuts in the skin, and it's been classified as a class 3 pathogen and potential biothreat. There are a number of clinical symptoms, including pneumonia, septicemia, right through to the chronic disease with symptoms very similar to cancer. And unfortunately, this disease is difficult to diagnose. It often is confused with tuberculosis, and its treatment is hampered by antibiotic resistance, which is seen not only for this bacterium but with bacteria, all bacterial pathogens in general. And of course, as with all drugs, they don't prevent a relapse. So this would be an ideal solution to this problem, would be the development of a vaccine, which has been deemed possible by experts in the field. So in this context, I was involved in an interdisciplinary project together with a number of collaborators from the UK and also in Italy, with the aim to try and use structural biology combined with in silico epitope mapping to try and re-engineer known antigens from Burkholderia pseudomallei and make them into better antigens. So the general workflow of the project is shown here and ranged from the antigen identification, which was largely based on known seroreactive antigens from the literature, right through to antigen design and production, 3D structure determination, and then in silico epitope mapping. So our work was largely based on the production of the antigens and 3D structure determination via X-ray crystallography, which I'm not going to go into detail about this technique as it's quite complicated. However, as its name suggests, you can imagine that it involves the production of protein crystals, which is the bottleneck in this procedure, and it's not always possible to grow protein crystals. We have a number of automated methods in the laboratory. We have 2 crystallization robots, which kind of helps optimize this possibility. So the heart of the project relies on the application of the methods, the in silico methods developed by Giorgio, that he will explain later, to the 3D structures that we generate. So the aim is to use his methods, Giorgio's methods, to try and identify the most immunoreactive portions of these antigens that are then designed as synthetic epitopes or recombinant domains for subsequent in vitro immunological validation to understand effectively if his methods were able to identify the best and most reactive regions of the protein. So this offers evident advantages of not requiring in vitro epitope mapping procedures. So I'm going to illustrate the very first test cases that were carried out on 2 seroreactive antigens that were specifically recognized by antibodies from seropositive melioidosis patients and also convalescent patients. So the idea was that since these are both convalescent antigens, perhaps they have some role in inducing immune protection. So in the top left panel, you can see the three-dimensional crystal structure of the first antigen, which is called PAL, for peptidoglycan-associated lipoprotein. And you can see in blue the epitope peptide that was predicted by Giorgio's method. So this epitope was then synthesized as a free peptide and used to induce antibodies that were then tested in a number of in vitro immunological tests by our collaborators in Exeter and Konkan University, so Richard Titball's group and also Ganjana Lertman-Monkolchai's group. As you can see in the top right panel, antibodies raised against this epitope were found to be able to induce antibody-mediated bacterial agglutination, which is one of the mechanisms of our body to clear the pathogen. And interestingly, this wasn't observed for antibodies that were raised against the whole entire recombinant antigen, so suggesting a first indication that, in fact, Giorgio's method had picked up an immunoreactive epitope that was with improved properties with regards to the recombinant antigen. In the middle panel, we can see an example of the immune seroreactivity test carried out with this peptide against different serogroups of patients, so negative, positive, and convalescent. And you can see it has quite a distinct recognition profile, and in particular was able to detect recovery antibodies, so also hinting at the use of this epitope for diagnostic purposes. And in a final experiment, the antibodies raised against this epitope were also found to have bactericidal properties in experiments using purified human neutrophils. So these antibodies enhanced bacterial killing by these neutrophils, and this also wasn't seen with antibodies raised against the recombinant antigen. So this was a good start. The same approach was applied to a second antigen, the flagellin antigen shown at the bottom. And the three epitopes picked up by Giorgio's methods are indicated in red, black, and green. And also they were found to have interesting and potential diagnostic and therapeutic potentials in these analogous in vitro immunological experiments. And also with two epitopes that were detected from this antigen, they were also shown to be able to stimulate cytokine production, and therefore there were also potential T cell epitopes, which is something that is particularly relevant to melioidosis, since being an intracellular pathogen, both arms of the immune system should be activated in an eventual vaccine. So our approaches were applied to other structures. We solved, I think, around 9 during the project, generating a large panel of epitope peptides that were used to be presented in peptide microarray format by our collaborators at the CNR, Marina Kretish's group. And this peptide microarray was successfully used to diagnose melioidosis, or Burkholderia pseudomallei infections, in comparison with healthy controls. And as an added value, the same peptides were also able to detect Burkholderia Chenochirpatra infections in patients with cystic fibrosis. I think someone's got their microphone on. It's kind of, anyway. Okay, so this was quite a nice finding because it showed that the Burkholderia pseudomallei peptides could also diagnose a similar but unrelated Burkholderia infection in a different clinical setting and also in another part of the world. So this project is already finished. These are some of the other antigens that for whom we solved the structure. So we have generated a lot of materials and epitope peptides for testing. And we have a present pump priming project that's ongoing in collaboration with Julian Cortazar, who's the PI of the project. And we actually just recently collected data for his antigen, so we're hoping that also this will be able to go into our epitope prediction pipeline. You can see there's question marks. You can see converting it into PowerPoint does something to the formatting, but never mind. So the approaches we use are general and can be applied to any pathogen, any protein antigen from any pathogen, which is something we're presently doing in an unrelated project where we're targeting diseases that are considered to be emerging in the Lombardy region. They're typically tropical diseases. For example, we're looking at protein antigens from the flaviviruses, dengue, Zika, chikungunya, and also the malarial parasite, and 2 human parasites, schistosoma and trypanosoma cruzi, which are responsible for schistosomiasis and Chagas disease, respectively. Unfortunately, these are diseases that are emerging in the Lombardy area due to increasing temperatures, climate change, and also the fact that it's one of the most affluent areas of Italy, so it's very attractive to incomers from other countries. So it's this project is still ongoing. We have an extension due to the lockdown. Things have been slowed up, but so far we have gathered a lot of materials also here, generated structures and epitope peptides. So in this case, the scope of the project is wider, and the aim is to develop a peptide-based microarray that presents multiple epitopes from multiple pathogens to be able to do a one-shot diagnostic test for multiple diseases. And in the project, we also have people who are working on actually developing the hardware to read the microchips. So I think the diagnostic potential of these epitopes is quite clear. There are also therapeutic potentials. However, all of you must know that peptides are not particularly good immunogens alone. They have short plasma half-lives, they're quite unstable. However, they can be easily conjugated to other molecules, and also there are a number of innovative strategies that can try and improve their presentation and therefore their immunogenicity. And that's something I'm going to talk about now. So the first is epitope grafting, which we carried out in collaboration once more with Giorgio's group. They also developed an epitope grafting software. So what is this? Well, it's basically the removal of an epitope from one antigen and its transplantation onto a second antigen, which presents a structurally suitable area to actually host the epitope. So for example, we took epitope 3 in red from the PAL antigen, and it's been transplanted onto the flagellin antigen, so it's also shown in red. And we did this in the opposite direction, removing the blue epitope here with joint T cell and B cell activities and transplanting it onto the PAL antigen. And the idea behind is by combining different epitopes from different antigens on the same scaffold, perhaps we could generate a kind of super antigen. So that's something we've been trying also with the third antigen, which was selected due to the fact it's a good B cell and T cell antigen, and also due to its particular structure, which you can see here. It's a U shape, and it's almost entirely helical, which lends itself very nicely to the transplantation of epitope 3 in different positions. So we can assess the different immunological effects depending on where the epitope is positioned. Furthermore, it's highly structurally stable, and it's also dimeric, so the transplanted epitope is present in double copy. And in blue, we have the actual own epitope of 2520, and of course this must be considered when you're carrying out epitope grafting. You must be careful not to overlap with that antigen, with that epitope. Another strategy to try and improve epitope presentation is on the surface of nanoparticles, which may be chemical, for example, gold nanoparticles, or also on the surface of biological vesicles. And that is also something that we're doing in collaboration with Guido Grandi's group at the University of Trento. They are particularly interested in the use of bacterial outer membrane vesicles for the presentation of epitopes, and particularly they work on cancer vaccines. So these vesicles are naturally produced by gram-negative bacteria, and they're very easily produced. They're just quite simply harvested from the culture media. They have an added advantage as vaccine delivery vessels, since they are naturally immunogenic. They contain a number of outer membrane proteins and also polysaccharides on their surfaces, and they can be easily decorated with epitopes and also entire proteins by genetically fusing them to an outer membrane protein that targets their transport to the surface of these vesicles. And actually, the vaccine against meningococcal B contains an outer membrane vesicle component. So in a previous Validate Pump Priming grant, in collaboration with Greg Bancroft's group, they carried out a pilot protection study using melioidosis mouse models, and we tested some of the outer membrane vesicle preparations that Guido's group prepared that presented the 3 flagellin epitopes I showed you in the previous slide, and also epitope 3 from PAL, and also the chimeric antigen on the surface. Unfortunately, despite the fact they all induced high antibody titers, we didn't actually see any long-term protection. However, there are a number of things we still have to try, for example, combinations of the different epitopes together. We also have other grafts and other epitopes to try, so this is something that could be done in the future. So I wanted to keep it short and quite general because I know we have a varied audience. I hope I managed to show you how structural biology can be combined with computational biology to try and identify the most immunoreactive portion of a protein that can then guide the design of antigenic molecules or epitopes that can serve as diagnostic biomarkers. And one can think in the future, if you have a microarray that presents epitopes belonging to different infection phases, also it can help diagnose the progression of the disease and to help tailor the treatment of the disease. And in our READY project, we're hopefully aiming to try and develop a microarray that can detect multiple diseases in one shot by presenting epitopes belonging to different pathogens. We also showed that when you remove the epitope from its recombinant antigen, it can be endowed with improved and different immunological properties in comparison to the starting antigen. So even if you have an average recombinant antigen, there is still possibility to improve it and make it a better antigen. And epitopes may be easily presented in different platforms, such as epitope grafted molecules, nanoparticles, and outer membrane vesicles for in vivo testing. Okay, and that's me done. I would just like to thank all the collaborators over the years from all over the world, in particular Giorgio's group, also Greg Bancroft's group, Guido Grandi's and Gan's group in Konkan, and of course the people I've worked with in Milan. Okay, and everybody for their funding, Validate, Diamond, and iNext, and also local and national funding. Thank you.
So I think we're going to hand over to Giorgio now, if you're ready, Giorgio. I think you need to stop. Oh, no, yeah.
I stopped sharing, yeah.
You have a mute, yeah.
So I'll start my video. Can you see the presentation all right?
That's working fine. Let us know, everyone, if you can't see the slides.
Okay. So thanks for having me today, and I'm very happy to be able to show you the results that we have obtained in the last few years with computational approaches. So in my talk, I wanted to just give you some sort of overview of the methods that we use for predicting immunoreactive patches on protein surfaces, starting from the crystallographic determination of protein structures by Louise and her co-workers. Then I will try to give you some examples of how we use these approaches to binding prediction, and then to the design of diagnostics that have already been introduced by Louise. So basically in the group we tend to use computational simulations, in particular molecular dynamics, which is a technique where you can simulate the motions of all the atoms under the determined by the effects of the chemistry of the surroundings of the atoms that will impose a force that will bring single atoms in a protein to move to a new position and generate this sort of jiggling and wiggling of the protein that will occur on several different time scales, from femtosecond to nanosecond to millisecond. So what we want to do from here, from this messy representation, is to extract the motions that combine and will determine the slow motions of the protein that are typically connected to the functional events or to functional duties. So one particular type of function is the recognition of another protein, like in protein-protein interactions, or the recognition of the antigen surface by antibodies, which is what we are talking about today. So our goals will be to discover functional spots by analyzing the main energetical determinants of stability and recognition in these proteins. So when we look at a protein antigen interacting or being presented to an antibody, what we want to find is, what is the most reactive or the region on the protein antigen that is most likely to interact with the antibody. And so we are interested in finding the epitope. And what we want to do also is to try and develop methods that will highlight or will single out the epitope region from the whole structure of the protein. So how do you do that? We basically start by implying that not all the interactions within a three-dimensional structure of a functional protein are the same, but rather a protein can be divided into different sectors. Some of these sectors will be devoted to protein stability, and the residues belonging to these sectors will share among them and with other residues very strong interactions, while other regions, in particular some of the regions on the surface, will be sharing a minimal amount of stabilization energy, and so they can change conformation. They are not blocked in one conformation by interacting with the core, and they can be given out to recognition by some other partners or some other proteins. So we call this the recognition sector. So what we need to do is to use the information in terms of structures, stabilities, and energies that we can calculate from our simulations in the molecular dynamics framework to basically subdivide the protein into the hotspots, the determinants of stability, and the energetically uncoupled regions or the soft spots, which are typically being uncoupled, being flexible, being potentially prone to mutations, because a mutation in one of these regions will not impact on the stability of the antigen and will thus not be detrimental to the function of the antigen. These are all hallmark properties of epitopes. And so they will be considered as the potential binding sites or antibody binding sites. So we find them by looking at the fully solvated MD simulation and then trying to compile a sort of energy matrix. So here on the two-axis you will see a residue number. So this is the sequence on the x-axis and this is the sequence on the y-axis. We simplify this matrix by eigenvalue decomposition, and then we filter it by the matrix of contacts within the protein, so which tells us which residues are close to which. And the final result is a matrix of where we have the information on which residue is close to which, and also how strong is the interaction between these connected networks. Then we pick out only the residues that are weakly or connected or decoupled with the rest, with respect to the rest of the protein. And as they are all very near to one another on the surface of the protein, they will form a continuous patch, which we call our epitope. So this is, the first case we applied the approach to, which is reported here pictorially, and here you have the sequence of the residues that are actually binding to the epitope, which was present to the antibody, which was present in the three-dimensional structure of this complex. And here in the predicted line, you have the sequence that we have predicted to be the epitope, and you see there's a good match between the two. Then we apply this type of approach to another series of different proteins for which we had the structure, the crystallographic structure in isolation, and also a crystallographic structure of the same protein in complex with an antibody. So we ran the prediction on the structure of the protein in isolation, and then we identify the epitopes, and we check which of the residues that we predict to be epitopes in epitopes are actually within contact distance with the antibody that is bound in the other structure. So we applied it to this zoology of proteins with different structures, different topologies, and so on. And generally, we are able to have a good predictor with an area under the curve of about 0.71. With good sensitivity, good specificity, and also good accuracy. So in collaboration with Xavier Daura from the University of Barcelona, the Autonomous University of Barcelona, we set up a server which is called Dept from binding epitope prediction from protein energetics, where you can just submit the PDB structure of your protein and you will get back an email with the epitope information, with the predicted epitope information. Then, in collaboration with Louise and the other collaborators that she mentioned, we started working on Burkholderia pseudomallei, and we wanted to see whether our predictions could be translated into actual usable molecules that you can also chemically modify. As we are chemists, we want to play with molecules, modify them, understand their structure, activity, and function relationships. So we started by one case, which Louise has briefly introduced, which is this PAL-BP protein, where we identified a potentially reactive region here in this alpha helix, which we synthesized as an isolated epitope and then presented it in ELISA plates to interaction with antibodies, showing that we could be able to distinguish between different patient groups, seropositive, recovered patients, in a way that is using a 10 and 15 residue small peptide that you can make in a couple of hours and purify in another couple of hours, having performances that are similar to using the whole PAL-BP protein, but with a much smaller molecule, which is much more manageable, less costly, and so on. We also use this small epitope to inject into rabbits to generate antibodies, which Louise has already shown were able to induce phagocytosis, induce oxidative burst, and induce bacterial killing in Burkholderia pseudomallei, and also to induce antibody-mediated, antibody-dependent agglutination as a first step to the elimination of the pathogen. And this is a control with E. coli, a negative control with E. coli, where you see that the epitope, the antibodies against epitope 3 work only on Burkholderia and not on Escherichia coli. Then we asked what could happen if, if we could block the structure of the peptide in a structure that is very similar in isolation to the one that the peptide would have in the context of its cognate protein. So we developed some chemical approaches based basically on click chemistry to block this conformation of the isolated peptide. You know that isolated peptides in solution do not really have a folded structure. to force the folding of the epitope mimic into an alpha helical structure. This is the CD spectrum showing that. So this is the synthetic line of the signal of the synthetic epitope, which is completely alpha helical. This is the PAL3 epitope that is non-structured, and you can see that in all cases the PAL3H, so the helical peptide, the blocked helical peptide, has got some better diagnostic and predictive features, better than the ones of the original peptide. Then we extended it to develop small microarrays recapitulating the reactivities of versus antibodies of many proteins. And we also developed in parallel some chemistry to be able to block the epitopes, the epitope mimics on the surface of the microarray to optimally present it to the interaction with an antibody. So with our chemistry, these types of synthetic epitopes are optimally oriented for interaction with the antibody. They are not in contact with the surface, but they mimic solution-like conditions. So we apply this thing to these concepts to several proteins whose structures have been solved by Louise, and also to some homologous proteins from other Burkholderia species. And we developed an antibody with, a microarray plate with different peptides. So here you can see that some of the peptides, when they bind the antibody, which is then revealed by fluorescence, antibodies from the bloods of Burkholderia positive patients, only these ones are enlightened, show up, while healthy patients do not give any signal. And these sera belong to patients who have who are cystic fibrosis patients who have been infected by cystic fibrosis and by Burkholderia bacteria of the Burkholderia genus. And we also tested whether the same peptides could be recognized by other pulmonary bacteria that infected cystic fibrosis patients, and you can see that the response is much lower in the cases of the other bacteria compared to the ones from Burkholderia. So the idea is that we can build a sort of heat map to stratify the patients and the responses of the patients, to separate, to cluster up the Burkholderia infected patients from patients infected by other types of bacteria. So we have been extending this in the last couple of years with colleagues in my institute and with Louise in terms of better presenting the epitopes, in terms of improving the chemistry, improving the designs, and also introducing some sort of better control on the conditions of epitope synthesis. Finally, we tried to extend this approach, the computational approach, to cover also some cases where the protein could be covered by glycans, which is a strategy that microorganisms or viruses sometimes use to hide epitopes from the presentation to the surface, to the immune system. And we applied this approach first to the spike protein of SARS-CoV-2 virus, which is completely covered in glycans, as you can see here in this slide. And what we did was to include the carbohydrates in our program, to remodel our program to include carbohydrates, and also to remodel our program to deal with multi-domain proteins. So not only do we find short sequences that are decoupled from the rest of the protein, but now we also can find domains which are decoupled from the core of the protein in a multi-domain protein, and decoupled domains might be the most immunogenic ones that you can just cut off the protein and then use for immunization, or as smaller domains rather than using a big molecule. And in the case of the spike protein, you can see that the domains that come up that emerge as immunogenic, most immunogenic, are the RBD domain, the receptor binding domain, which is the one used for vaccine development and for antibody development, for instance, by the Regeneron antibodies. The N-terminal domain in some regions here, which are actually hidden by sugars, by carbohydrates. And then we also use the more restrictive definition of the epitopes to look at smaller sequences. And here you can see the sequences that we predict in brain superimposed to the crystal structure of antibodies binding to the spike protein in the N-terminal, and in the and also we have examples on the RBD domain. So we also have extended this to glycosylated proteins. So the computational approach is of completely general use. So in conclusion, I hope I have shown you that we can proficiently use computational methods for molecular recognition. We can design immunoreactive molecules based on rational identification of reactive regions. We can combine computational and bioorganic chemistry to investigate molecular recognition in immune systems. And also we can develop chemical probes to probe antibodies and the responses. And then we have made progress in the design of immune diagnostic tools and in the design of biosensors. So I would like to thank you for your attention, and also thank my collaborators Claudio Peri, Riccardo Capelli, Filippo Marchetti, and Stefano Serratian, who have worked with me on these projects, and of course Louise and Martino Bolognesi for their super precious collaboration with the crystals and discussions and patients, and other collaborators like Richard Tipol and Gan for their help with the validation of our predictions. And thank you all for your attention.
Thank you very much, Giorgio and Louise. So we're going to move on to the Q&A session. You should all theoretically be able to unmute yourselves now. I had to turn it off because somebody kept unmuting themselves for some reason, but that should all be working now. If you have any questions, click on the raise your hand button like I just did then, or type your question into the chat thing. So does anybody have any questions? This is the awkward bit where everyone's a bit scared to go first.
Hi Blakely, it's Adam. I'm afraid I can't see the raise hand, so I'll just raise my voice instead.
Just go ahead, yeah.
Really interesting talks, thanks. I've got a question for Louise, actually. It's hi Louise.
Hi.
Again, really interesting talk. I was particularly interested in the vaccine work you were involved with Greg Bancroft with the OMVs. You mentioned that you had good antibody responses, but unfortunately you didn't see any protection.
Yeah.
Was there any measure of T-cell immunity?
No, we didn't. That actually was one of the aims. That was supposed to be one of the aims as well of the project, but we had some problems at the beginning, and so we just ran out of time. But yeah, that would be something we would like to look at.
Yeah.
Sorry.
I thought I'd be polite and say hello. No, it's an area that I've always been interested in, and one potential concern when I've been involved in some vaccine work with melioidosis, a lot of the targets of the antigens come from serological sort of screens. So we know they're dominant, and there doesn't seem to be a lot of work that's been done using sort of unbiased screens for T-cell dominant antigens. And I wonder if the sort of lack of T-cell immunity, where we've struggled to see it, whether it's a consequence of our sort of vaccine delivery systems, or whether we're looking at the wrong antigens. But
yeah, something we also have noticed is what you see in vitro doesn't necessarily reflect what you're going to see in vivo. It was quite disappointing. We were so kind of looking forward to positive results, but it wasn't to be.
Interesting work though.
Thanks.